So this is the last chunk of lecture 14. It's also the last lecture chunk we will spend on the citric acid cycle. Feel free to pause the lecture for a moment or two if you need to grab a tissue and wipe your eye. I know if you're half as sad as I am to see the citric acid cycle go, you'll definitely need a moment to collect yourself emotionally. I'm going to plow through, though, and say that this is the end of chapter 19, as well as the end of the citric acid cycle material. We've already talked about the second half of the citric acid cycle itself, as well as its regulation, and so here we'll have that discussion I've been promising on the role of citric acid cycle from a bigger view on metabolism overall. So let's have that conversation. Let's start very broadly and just realize that we eat everything. If it has lived at one time, there's someone on the planet who eats it. Essentially what that means is that we are constantly getting from our diet not only sugars, but also proteins and fats. Those are the three biomolecules that we essentially consume. They're the three types of biomolecules that essentially represent everything that's in a living cell. So when we eat sugars, proteins, and fats. First, these large polymers are broken down into their smaller and simpler subunits. That's the large scale of metabolism that largely happens in the small intestine. Then those subunits, those monomers, are further metabolized at the cellular level. And they can all be converted to acetyl-CoA. We talked about that a little bit in the last chunk. And once converted into acetyl-CoA, they can all enter the citric acid cycle. So let's see that. This is the road that we've been on all along. We've eaten carbohydrates. Glucose is what we've been using for our example. And through glycolysis, we convert that glucose into phosphoenol pyruvate and then pyruvate. Pyruvate is then turned into acetyl-CoA through the action of the PDC, and acetyl-CoA is fed into the citric acid cycle as we've discussed. We harvest electrons along the way, and we'll pick up with those electrons in the next lecture. But look what else can become pyruvate. Threonine, serine, cysteine, glycine, alanine. These amino acids can also become pyruvate, which can be converted into acetyl-CoA by the PDC. Those are amino acids coming from proteins that we eat. Look what other amino acids are possible. Isoleucine, leucine, tyrosine, lysine, phenylalanine, tryptophan. These can be converted to acetyl-CoA directly, independent of pyruvate, and also fed into the citric acid cycle. And as I've already said, lipids or fats can be broken down into acetyl-CoA through beta oxidation. So we can feed almost any biomolecule possible into the citric acid cycle by converting it either into acetyl-CoA directly, or converting it into pyruvate, which the PDC can then turn into acetyl-CoA. But let's look a little closer. Some molecules that we consume, some amino acids in particular, can be converted not into acetyl-CoA or pyruvate, but instead be converted into citric acid cycle intermediates themselves. Asparagine and aspartate can be converted into oxaloacetate. Oxaloacetate can actually be back converted into malate as well. So we can turn these amino acids into citric acid cycle intermediates. Tyrosine and phenylalanine don't necessarily have to become acetyl-CoA. They can be converted into fumarate. Glutamate, proline, arginine, glutamine, histidine can all become alpha-ketoglutarate. And threonine, valine, isoleucine, and methionine can be converted into succinyl-CoA. So we can make citric acid cycle intermediates by using the amino acids in the foods we consume. Doing so can actually speed up metabolism. Let's consider that for a minute. How could making citric acid cycle intermediates speed up metabolism? Keep in mind that the citric acid cycle is not limited by enzymes. Enzymes are very quick to act and they recycle. A single enzyme can produce or catalyze the same reaction many, many times per minute. What limits the citric acid cycle speed is the intermediates that we have. If we have one oxaloacetate, that oxaloacetate can be turned into citrate through step 1, and then citrate synthase would sit there idle, waiting as step 2 occurred, and then step 3 occurred, and then step 4 occurred. It's actually the availability of either acetyl-CoA or oxaloacetate that slows the citric acid cycle down. The enzyme is idle while one round of the citric acid cycle goes through. Now let's consider that a little bit further. If we have an abundant amount of acetyl-CoA on hand, One acetyl-CoA will enter citrate synthase for step 1, be linked to our 1-oxaloacetate and our 1-oxaloacetate intermediate, and then both will remain idle while the citric acid cycle cycles. All of that acetyl-CoA is not useful to us because there's no oxaloacetate around to link it to. And the enzyme is remaining idle because without another set of reactants to work on, product can't be made. What this means is that the citric acid cycle can be sped up not by speeding up the individual steps of the cycle, but instead by making it possible to run many cycles simultaneously. If we had one citrate synthase enzyme, but an abundant amount of acetyl-CoA and an abundant amount of oxaloacetate, that enzyme could quickly condense one acetyl onto each oxaloacetate in a row, making many citrates, and then isocitrate isomerase could convert that into isocitrate, and then we could do our oxidative decarboxylation. So, in fact, the more intermediates of the citric acid cycle, the more oxaloacetate, the more succinyl-CoA, the more fumarate, the more malate, the more intermediates we have, the more citric acid cycles we can have running in parallel to each other, the more electrons we will harvest per unit time. So the ability to make citric acid cycle intermediates is a very powerful ability for the cell to have, because by making more intermediates, by making more oxaloacetates, we are making more citric acid cycles. The number of oxaloacetates in a cell dictates the number of simultaneous citric acid cycles the cell can have running. That's key. And so we can make citric acid cycle intermediates by using these types of catabolism pathways. Asparagine and aspartate can all be used to make oxaloacetate itself. And the more oxaloacetate we have, the more citric acid cycles we have running simultaneously. But let's not focus just on catabolism. Let's talk about making molecules as well. I've mentioned a few times now that the citric acid cycle is a major player in anabolism, in making biomolecules. But if we're going to make things from the citric acid cycle, that means we're going to have to steal intermediates away from the citric acid cycle. If we steal too many intermediates, we run the risk of shutting the citric acid cycles down completely, and that's going to mean certain death for the cell. The cell will not be able to make enough ATP without the citric acid cycle running. So the citric acid cycle must have a mechanism by which it can be replenished, and we've just seen one way that we can do that. We can take aspartate and other amino acids that we've consumed and use them to make oxaloacetate, and once we have oxaloacetate on hand, we have ourselves a citric acid cycle. So we can replenish oxaloacetate through aspartate. But we can replenish oxaloacetate another way as well, in an ingenious way. There's an enzyme that we'll talk about in a couple lectures called pyruvate carboxylase. And pyruvate carboxylase can make oxaloacetate from pyruvate. Now think about this for a moment. If glycolysis is working fine, we're making plenty of pyruvate. That pyruvate can be converted into acetyl-CoA by the PDC. But if there's no oxaloacetate around, that acetyl-CoA has nowhere to go. Remember, high levels of acetyl-CoA inhibit the PDC. So if acetyl-CoA starts to build up because there are no citric acid cycles for it to join, no oxaloacetate for it to condense with, the PDC will shut down and pyruvate levels will climb. Once pyruvate levels get high enough, pyruvate carboxylase will act on that pyruvate and make it into oxaloacetate. Isn't that amazing? And that oxaloacetate will then be available to condense with acetyl, and the citric acid cycles will resume. So pyruvate can fix a missing oxaloacetate situation by being converted into oxaloacetate itself. This is a little bit of a preview of a slide that I'm going to show you in a couple lectures. We're going to talk about a process called gluconeogenesis, which essentially breaks down to meaning making glucose from scratch. I won't go into the details here, but I want to show you that this step I'm referring to is actually part of this process. If you look down here on this slide, you'll see that pyruvate and carbon dioxide can be brought together in a carboxylation reaction to make oxaloacetate. That's right here. Pyruvate, a little bit of energy in water and carbon dioxide, can make oxaloacetate for us. It's energetically costly, but it's better than dying. Pyruvate carboxylase catalyzes this reaction, and this is the oxaloacetate that we can use to replenish our citric acid cycles. Once we can make oxaloacetate from pyruvate, as we've just described, this allows us to replenish the citric acid cycle and get it up and running again. So we can steal from the citric acid cycle for anabolism. We don't need to worry about shutting the citric acid cycle down as we steal intermediates from it, because we have this fallback strategy for creating citric acid cycles from scratch, for using excess pyruvate to bear or make new citric acid cycles. Just as we can break fats and lipids down into acetyl-CoA, acetyl-CoA is also used, excess acetyl-CoA is also used to build fats. So if we're talking about anabolism, we're talking about making anything, including some things we might not necessarily want. So excess acetyl-CoA is derived from the consumption of excess sugars. If we have too many sugars on hand, if we continue to consume sugar even when our ATP levels are high, we will build up acetyl-CoA levels. And that acetyl-CoA will be used to make lipids. Remember, it's glucose to pyruvate in glycolysis, pyruvate to acetyl-CoA by the PDC, so high levels of glucose, too high levels of glucose, can easily result in high levels of acetyl-CoA. It always traces back to sugars. Now we can convert lipids to acetyl-CoA. We can convert lipids to a source of energy, but we can't convert lipids to carbohydrates. We don't have enzymes to do that for us. Once we make lipids, they're stuck, or they're broken down by beta oxidation. But I'm showing you here that we can convert carbs to fats. Glucose can be converted to pyruvate, pyruvate to acetyl-CoA, and acetyl-CoA can be used to make fat. So excess carbohydrates can create fat. And I don't think you needed me to tell you that. We see here is now a fairly outdated map of obesity in our country. These percentages that you see here are percentages of obese individuals in the United States. We see that everywhere in our country, at least 15 to 20% of the population is obese. Most of the country, 20 to 25% of the population is obese, and far too large of a section of our nation, over a quarter of the individuals, are obese. These numbers are not due to high-fat diets. That's commonly thought, but it's not the case. The number one contributor of obesity in our country is actually high-fructose corn syrup-containing products, most notably sodas. Too much sugar is creating too much fat. And too much fat is a health epidemic in this country. It's a real shame that too many people are under the guise that if they just eat a low-fat diet, they'll be protected from obesity. Glucose can be converted into fat, and I've just shown you how, by excess acetyl-CoA levels creating too much lipid formation. But we can not only use pyruvate to make oxaloacetate, we can not only use acetyl-CoA to make fats, we can also use intermediates of the citric acid cycle itself as precursors for amino acid biosynthesis. Oxaloacetate can be used as a precursor to make aspartate in a single reaction. And then that aspartate can go on and be used to make a lot of different amino acids, such as asparagine, methionine, lysine, threonine. And threonine itself can be converted to isoleucine. So that's 6 amino acids that can directly trace their lineage back to oxaloacetate. Alpha-ketoglutarate can be used to make glutamate, again in a single reaction. And glutamate can be used to make many of the remaining amino acids we didn't just cover, including glutamine, proline, and arginine. That's 4 amino acids from alpha-ketoglutarate. 3-phosphoglycerate, that's 3-PG, our old buddy from the latter stages of glycolysis, can be converted into serine, and serine can be used to make cysteine and glycine. Pyruvate itself can be used to make alanine, valine, and leucine. Succinyl-CoA is the primary precursor for making heme. the oxygen-carrying component of our hemoglobin in our red blood cells. It is just insane how important the citric acid cycle is to everything we think of as life. In a nutshell, if we think anabolically, these are the molecules that come out of the citric acid cycle. We see some recaps of what I've already said. Acetyl-CoA can be used to make fats. It's also playing a major role in the production of leucine and lysine. Pyruvate can be used to make a large number of amino acids. Alpha-ketoglutarate, glutamate and the amino acids we can make from it. Succinyl-CoA, porphyrins. Porphyrin rings are the structural components of heme. Malate can be used to make oxaloacetate, not only amino acids from oxaloacetate, but also our nitrogenous bases, the pyrimidines. And phosphoenol pyruvate can be used to make carbs through gluconeogenesis, as well as an array of other amino acids. This is why I and most other biochemistry professors call the citric acid cycle the hub of metabolism. It is the most central process in all of cellular metabolism. This beautiful beast, I get chills every time I see it, represents the entirety of the metabolic activities of a eukaryotic cell. Every metabolic reaction that goes on in a eukaryotic cell is represented on this diagram. If you're curious, you can find these online at a higher resolution, or I can certainly provide you with one. In graduate school, there used to be a poster form of this on one of the walls in one of the graduate classrooms, and I used to, this is how much of a geek I am, I guess, but I used to love just looking at it and just appreciating how complex this process was, and then having that moment of awe that despite all of this complexity, we as a people understood it and actually figured it all out on our own. That's really something to stand in awe of. But anyway, not to digress, if we look down this central corridor of this map, we see some old familiar faces. We see glucose here, entering this, converted into glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bis phosphate, 3-PG, 1,3-BPG, 2-PG, phosphoenol pyruvate, and pyruvate. This is glycolysis running down the center of this poster. The PDC converts that pyruvate into acetyl-CoA, and that acetyl-CoA goes into the citric acid cycle. Notice the position of the citric acid cycle on this map. It is directly in the center. It is in the center of this central line. It's in the center of the poster. And just take a moment to appreciate how many things feed into and feed out of this central hub. The citric acid cycle is the unifying hub. It is the keystone component of cellular metabolism. That's why we spend so much time on it. That's why we give it such due deference. It is just an amazingly important component to what keeps us alive. And it doesn't matter what version of a metabolic map you look at. Look for the large circle in the center of that map, and it will undoubtedly be the citric acid cycle, representing the importance of this process to a cell. It's important. Brief point before we end this. Why is the citric acid cycle part of aerobic respiration if we never saw molecular oxygen show its face so far? Well, yes, it's true. Oxygen is not required for any of the reactions of the citric acid cycle. In and of itself, the citric acid cycle is an anaerobic process. It doesn't require oxygen. Yet it is the defining pathway of aerobic respiration. What gives? Well, as we'll see very shortly in the next lecture, everything done in the citric acid cycle, and specifically what I'm referring to as the harvesting of electrons, was done so that ATP could be made. And that ATP production with electrons requires oxygen. We'll see the reasons in the next lecture. Therefore, if we don't have oxygen available, we can't use the electrons we just harvested in the citric acid cycle to make ATP. And therefore, the citric acid cycle would have been useless. So even though the citric acid cycle doesn't require oxygen itself, the reason why we ran the citric acid cycle in the first place does require oxygen, and so this is indeed part of aerobic respiration. To summarize this last chunk, many different subunits of what we eat, many different monomers of the sugars, fats and proteins that we consume can be converted into acetyl-CoA and fed into the citric acid cycle. Some amino acids can also be converted into citric acid cycle intermediates, allowing us to replenish the citric acid cycle. Most importantly, pyruvate carboxylase allows us to make oxaloacetate directly from pyruvate, allowing us to have many, many more citric acid cycles running per unit time. Oxaloacetate can be made into aspartate, and aspartate can make many amino acids from itself. This gets at the citric acid cycle's role in anabolism. Alpha-ketoglutarate can be made into glutamate by a single reaction, and just like aspartate, we can make many amino acids from that. And so the citric acid cycle is playing a very, very large role in not only the production of amino acids, but also in the production of heme, of the nitrogenous bases of the purines and pyrimidines, and essentially of almost anything else you can think of that's important to a cell. So get that old dirty tissue ready again, because here comes the waterworks. We are saying goodbye to the citric acid cycle, and we will now move on to see what happens to all of those electrons we've been working so hard to harvest.